I wanted to thank Dr. Roos, Dr. Kissel, I'm Dr. Johnson, for joining us here. Really, Dr. Roos and myself were editors for a new journal, Neurology Genetics, which is a journal exclusively devoted to genetic diseases of the nervous system. It's now on PubMed Central, and it's an open access, and we're certainly looking for submissions. But that's not why we're here today. We're here to learn about genetic therapies for nerve and muscle diseases. And so with us we have Dr. Kissel, who's a world expert in neuromuscular diseases, and we're going to talk about some of the new therapeutic approaches in nerve and muscle disease. So with that, I'll turn it over to Dr. Roos.
So, John, before we talk about the new therapies on the horizon for SMA, maybe you could tell us a little bit about what set the stage for this that kind of led us into the possibility, what kinds of reagents, models, etc.
Well, that's when I give talks on SMA, one of the things I often say at the beginning is, or the subtitle to my talk is SMA from ugly duckling to swan, because I like to say that SMA was a disease that I think most of us didn't pay any attention to. Even when I was a fellow, it was just considered one of those typical rare, unusual diseases that we couldn't cure, and we just sort of told people to go home and live with your disease and die. And what really changed things dramatically and fundamentally was after the discovery of the gene in 1995, and oh, by the way, the disease is due to a mutation in the SMN gene, or the survival of motor neuron gene. It's an autosomal recessive disorder. So every patient with SMA has a homozygous deletion, or sometimes point mutation, in both copies of their SMN gene. But what really, I think, woke people up is, I like to laugh that when a gene is discovered, the first thing the gene jockeys do is they knock it out of a poor mouse and see what happens to the mouse. And when they did this, it was a lethal mutation, and all the mice were born dead. It was a lethal mutation. So they realized that something else had to be going on, and what that realization was that humans have a spare copy of this gene that's called the SMN2 gene, which is centromeric to the SMN1 gene. I explain it to patients, it's like having one of those little lousy spare tires on your car that'll get you 50 miles. And so that all patients with SMA, as I said, have a homozygous deletion or mutation in SMN1. But the SMN2 is very similar to the SMN1, except for a single base pair substitution, which results in the message that generates missing exon 7. So, but because of alternative splicing, the spare copy of the gene can make about 10% of the normal protein. So to summarize about a decade of intense genetic work, what we finally discovered was that the reason why mice, it's a lethal mutation, is they don't have any SMN2, whereas humans have SMN2. And the more copies of SMN2 you have, the milder your phenotype. Because of the alternative splicing, the spare copy of the gene makes about 10% of normal protein. And that immediately suggested several ways to go about trying to treat the disease.
I mean, so you summarized about a decade of work, but we don't have a decade worth of treatments. I mean, are there ongoing treatment trials, or what are the therapeutic genetic strategies that you could use?
Well, actually, we have been trying therapeutic strategies. So as I said, all patients with SMA produce some SMN2. And as with most, again, genetic disorders, when that was discovered, then Arthur Burgess, actually at Ohio State, his lab, was able to basically create transgenes where you put SMN2 into the mice so that it was no longer a lethal mutation. And if you put enough copies of the SMN2 in, it would actually result in prolonged survival of the mouse. So what that suggested immediately, and I can remember very well Arthur saying this to me, was that he said the nice thing about this work is that we don't even have to understand all this very well before we can come up with therapeutic strategies. And so the first strategy, which was the obvious one, was to try to come up with a drug that could increase the production of the spare copy of the gene. So if that copy made, I said 10%, and you need, let's say, 40 or 50% for normal motor nerve function, if we could come up with a drug that could increase SMN2 production, we might be able to correct the phenotype, and that's actually what I've been involved with. Unfortunately, those drug trials haven't been as productive as we might have hoped, and so more recently, two other strategies have been employed. One is to use what's called an antisense oligonucleotide, and again, the way I explain it to patients is to sort of trick the SMN2 into functioning like SMN1, so that the antisense oligonucleotide actually puts the exon 7 back in the message and produces full-length SMN. And there was just a publication in Neurology this year on the preliminary results of that. And then the third strategy, of course, is to quote simply, close quote, replace the gene altogether, which is ongoing at Ohio State with a trial by Jerry Mendel's group at Nationwide Children's.
So, John, there was this decade in which the genetics and models were investigated, and now there are 3 trials. I want to ask, do things look promising? And here we have a disease that was a completely hopeless one when you got into neurology. What is this going to do for the field, to neurology?
Well, that's what I started by saying that this disease has gone from ugly duckling to swan. And one of the things that these discoveries, because there's no other disease in humans that are like this, that have the spare copy of the gene, but it's made SMA this unbelievably attractive disease for researchers and people working in the field. And I think that we think this is the beginning of the end. The gene therapy trial at Nationwide Children's has now enrolled 50, and I'm a co-investigator on that, but we've now enrolled 15 patients. And at a follow-up for the first babies at 22 months, we've had no deaths in the type 1 babies. Now, to compare that, the valproic acid study in type 1 babies that we did along with the Project Cure group, and Dr. Raymond is in the audience today, was heavily involved with that. We had, I think, 40 patients, and we had, I think, 14 deaths within the 2 years that we enrolled in the study. So, and that's, other data have suggested that. So we've had no deaths, and I think what's even more impressive is that there are babies that are actually gaining milestones as opposed to losing them, which in the old days was just unheard of. So we're all thinking that this may be the beginning of the end. Neurologists, and particularly neuromuscular specialists, never use the C word, cure, but I think that's one thing that we're starting to talk about.
So, for example, you might imagine gene therapy or these antisense oligonucleotide approaches. It might be kind of a plug-and-play type of strategy. What's your experience with some of these other inherited muscle diseases like Duchenne or myotonic dystrophy? Do you think there's the same promise in those conditions?
I think it's yes. The answer to your question is yes. I think the outlook, because of the nature of the genetics in SMA, I think the outlook is probably not quite as rosy soon. But I think the whole antisense oligonucleotide, and now with CRISPR, the Cas9 technology, I think the ability to modify fundamental genetic defects is just astonishing. Probably the best example of that from the myotonic world is myotonic muscular dystrophy type 1, which is caused by a CTG repeat expansion in the myotonin protein kinase gene, which results in the message having these big CUG RNA loops. And again, as I tell my patients, Ionis, formerly ISIS, drug company now has a trial going on that we're participating in at Ohio State. And what the antisense oligonucleotide basically does is go in and, like a little pair of scissors, and activates an RNA nuclease that cuts this and results in the RNA then being digested and eliminated, correcting the downstream defects in myotonic dystrophy. So we're actually in the phase 1 trial of that, and too soon to talk about results, but I think the approach and the methodology is very exciting.
So, John, Are there barriers, do you think, that we're going to learn about? You know, for example, modifiers of the gene, and that maybe we don't know as much about SMA as we're going to learn from these clinical trials. And what's the future? Let's say one of these SMA clinical trials is successful. What do you think of as the next step?
Well, that's a great question, and the obvious one in terms of SMA is early detection. When you use this technology with gene transfer in the SMA mouse model, the mice die at 16 days, and if you give them an injection of the AAV9 self-complementary AAV9 virus with the gene on it, it's essentially curative, and I like to joke that the mice die of old age rather than dying at 16 days. If you give it at 5 days, again, about a third of their lifespan, there's a partial effect. But if you give it at day 10 or 11, there's no effect at all. So one big challenge is going to be to treat all of these patients early. So early identification is going to be key, probably with perinatal or prenatal screening. And another barrier, you know, I think the best example of this, when it relates to Duchenne dystrophy. And again, Jerry Mandel at Nationwide Children's did one of the first. It was a microdystrophin gene modifying, where they put a truncated form of the dystrophin gene back. And the thing that struck me, that if you think about it, it's so obvious. And actually, I told you I didn't prepare for this talk, but I did look up one thing, which was I couldn't remember how they had administered it, and it was intravenous. But this was published in the New England Journal. But one of the things that people forget is that if you're deficient in a protein, so if you've never made SMN protein, or if you've never made dystrophin for a Duchenne boy and you replace it, the body will mount an immune system. So in the initial study for Duchenne that was published, the boys actually had a T-cell response against dystrophin, partially because some of the exons they replaced were foreign to the child, but also because dystrophin's the largest gene in the body, so you can't put the whole thing back. So they tried to put a truncated form in, but when you put that in, the different folding of the protein creates epitopes that generates antibody and T-cell responses. So those are the kind of unexpected things that we're gonna face. And then also just the one other one is delivery. Clearly, you have to be able to get any type of gene therapy exactly where it's supposed to go. So for SMA, for example, right now, there is a sort of a mini debate in the field going whether peripheral infusion is going to be good enough or will it be enhanced by intrathecal administration, or both? And there's some evidence in animals for all three of those.
I think, you know, just to get after one of the barriers, one of the other issues in some of these progressive conditions is the age of diagnosis or being able to detect it early. Any other thoughts about how that will play into some of these treatments?
Oh yeah, I think that is an idea whose time has come. There's already several years ago there was sort of a mini debate in the SMA field about doing perinatal screening and screening these babies right after they're born. I think that with things like gene therapy, that's something that's just going to have to come. Up to this point, it's been sort of a catch-22 because people tend to not go for perinatal screening unless there's something you can do about the disease, like phenylketonuria. But I think that as these therapies get developed, I think the move to treat earlier and earlier is going to be a requirement. And there's even some evidence for that in the gene therapy trial being done right now. There's some evidence that the earlier babies that are treated do better.
So if one of the clinical trials is successful, what more has to be done before, for example, FDA approval? Are you thinking about those kinds of future issues?
Well, again, I'm just a co-investigator on this study, and Jerry at Nationwide Children and others that are doing this kind of work are pursuing this. I can tell you, I've been in front of the FDA for these studies once. It's very much debated within the field, and just a couple of issues. I mean, is it ethical now to do a placebo-controlled gene therapy trial in a disease that's invariably fatal, or type 1 SMA? That's one of the issues. Dosing, those issues come out. So those are all being worked out now for the next phase of the study, and age of treatment.
So I think those are our questions that we had for you, and I just wanted to say, just as a reminder, that Neurology Genetics is a new journal. It's been available about 12 months, now available on PubMed Central. Is really is an open access home for any of this work, or any work that involves the genetics of the nervous system. So with that, I think we have time for a few questions if anybody has any.
I just make. While people are thinking of their questions, but I actually did want to make just one last comment, which is, when we think of gene modification therapy, or quote gene therapy, we tend to always think in terms of replacing the missing gene or correcting the defect in the gene. But the term or the approach of genetic therapy can be even wider than that. For example, there are attempts now to modify the follistatin pathway. Follistatin suppresses myostatin, which inhibits muscle growth. So if you get rid of that, the myostatin will make muscles bigger. So there are trials going on now with antisense oligonucleotide to try to suppress that pathway to make muscles grow bigger in diseases like FSH dystrophy, where the molecular genetics is much more complicated.
John, while we're waiting for questions, how about international efforts with respect to SMA? Are there comparable kinds of trials going on outside the United States?
Actually, that's a good question. I don't know of any. Right now, Ionis with the SMA trial, I know the Europeans have been very involved. It's a very collaborative community, and I think that's going to do nothing but help, because every time there's a problem, I think there's a number of groups working to work this out. I tend to have a very skewed perspective, of course, because I've worked with Arthur Burgess and Jerry and the groups in Columbus, but there are certainly people all over the world working on this that have contributed to this, particularly in SMA.